and thanks also for the opportunity to speak to you all today about, as Margaret said, my key message that I want to get across really follows on from Bob's presentation about the possibilities that human tissue offer, and to perhaps get away from the mindset that human tissues in research mean fixed, frozen, essentially dead tissues, and that living human tissues can actually be used to really predict how a drug is going to behave in the clinic and eventually in patients. Now, I won't really spend too long on this. I'm sure you're all aware of the problems that the drug industry has experienced. But equally, I would say there are many very good drugs that have been brought to the market. So there's obviously, there are problems as well as many useful assays that are out there. And I want to also bring that message through during the presentation. But nonetheless, there's an increasing awareness that, this is actually an article I think about some of Bob's research a few years ago, that drug developers will have to carry out tests on live organs. There's this increasing emphasis on the need for a living system, a functional tissue, to provide some prediction of drug effects. So if safety and efficacy are the problem, how can human tissue be used to address that? Well, I would argue that human tissue should become a key part of the strategy for this, for three main reasons. These compounds can improve predictability. They reduce the risk of a drug failing at the latest stage when the costs are greatest. So I think the strength of the argument comes not only from the reasons, the scientific reasons and the reasons for testing on animals and so on. It's an extremely strong argument, but I think there are also very good commercial arguments. And when you tie that in with the benefits to patients, you have a number of reasons for using human tissue much earlier. So using it earlier means that if a drug is going to fail, because it will fail at some point, if it reaches the clinic and adverse effects are uncovered, or it simply just isn't efficacious, then it's going to fail anyway. So it's better for it to fail much faster and earlier in the process. So if we go on the premise, then I'll hopefully make a further arguments for why testing in tissue is valuable later on. But if we accept this statement, why is it not used more frequently? This it slightly touches on what Bob was saying. The first point, clearly, accessing tissue is difficult and expensive. What I want to say here is that it's very important in terms of taking human tissue research forward as an area of research that both academic and commercial research groups work together. Because I think this will also relate, I'm sure, to what Vivian says later on about public perception, is that the public really perceives breakthroughs in research as coming from academia, which is true. There are very many discoveries and research. We'll hear more, for example, from Kelly later on, who's developing new methods. But it's probably less appreciative of the joint efforts of commercial and academic groups to bring drugs to the market. And if we actually want to bring new medicines to patients, it requires a combined effort from both of those groups. So if the tissue is made more available and less expensive, I think that will feed through, first of all, to an increase in academic research. That's probably a major barrier for many academics, and why they often use animal models is the sheer expense of writing into grant applications. So if it comes through from that end, that will encourage greater use of human tissue models at the commercial stages that may follow on from that once a drug target is identified and taken forward to the chemistry and identification of an actual drug molecule. The second thing that we, as a company that does human tissue testing on a daily basis for pharmaceutical companies, and what we do is provide predictive data of how their drugs are going to behave in humans. What we quite often come up against is, I don't want to do that because human responses are too variable. And our response to that is that, well, that variation is going to exist in the clinical trials. And we do sometimes see this as a head in the sand phenomenon, in that this is more so in smaller companies that may want to just take a drug always to the next stage, is that they actually would prefer to use a very reproducible animal model, even if it's not necessarily giving the right answer. So I think there's has to be a change in mindset to see, this again comes back to having living, functional human tissues, is that these functioning tissues can be considered as a mini clinical trial. And that variation that exists in the patient population is actually a good thing. It can actually be put to use as long as the patient information and the results are considered in light of the patient information when the report is produced. The third factor is that current methods are accepted. There are thousands of tests done in the industry and academia every year. So changing this is difficult and it's a very long-term process, even if there are flaws in the method. But I would put alongside this statement that the current methods do produce useful information. I mean here all of the available tests. What we have to do, I think, to make human tissue more accepted is look at the arguments for using human tissue on a case-by-case basis. So almost on an experiment-by-experiment basis, looking at a particular model and method and arguing, is that more predictive than the current test? Because that, I think, is the best way in which the science and the ethical reasons for using human tissue will come through. But having said all that, is it actually realistic? If we actually were to do a survey of the number of human tissue publications, and I've had a quick look myself at PubMed to see in a particular area how many animal tissue or cell-based publications are there versus human tissue-based publications, then it's clear that there's really a small number of human-based tests being conducted, and that can be translated into an approximate guess at how many human tissue tests have been done per annum. And I would say, based on figures that I've looked at from HES online, that we only use a tiny fraction of the available human tissue in research. And this is where I'm going to differ very slightly from what Bob says, in that I completely agree about transplant material is the best, it's the gold standard, if you like, in terms of the quality of the material, and that it's an untapped resource. But I also believe that surgical residual tissue, bearing in mind that of course we all accept that the patient is the number one priority far and away, and that the tissue very often is not suitable for research or needs to be retained for diagnostic purposes. Even given that, the number of surgical residual tissues that are simply incinerated is, I think, a real tragedy, because it represents such a valuable test system. And these are numbers that I just picked. There's a huge spreadsheet on this database with the number of individual procedures in each tissue type, and you can see that they're quite significant. And what I also wanted to point out, that because of the concerns obviously about pathology and the need for diagnosis, it's not only tissues that are generated through pathology departments, but 2007, there were over 32,000 cosmetic surgery procedures in the UK, including nearly 2,800 tummy tucks, which I would say almost every one of those pieces of tissue was incinerated. So I'm not saying it's easy to access. We know from experience it's absolutely not easy to access these tissues. What I would like to get across is that there is a valuable untapped resource for both academic and commercial researchers to develop new drugs. Now, coming back to the bar graph that I showed early in the presentation, you'll have seen that the major cause of failure now is lack of efficacy. So basically the models that are used to test whether a drug is effectively treating a disease, these are artificial animal models of, say, inflammatory bowel disease, they're not that predictive of the ultimate efficacy in patients. And so I would argue that, again, more use can be made of diseased tissues to actually model the disease. I mean, it sounds obvious, but it's not done that often. Here are some examples of areas where we've been asked to work in a particular therapeutic area. Again, the choice of tissues really lead to the therapeutic area. So if you're wanting to study hypertension, then you investigate blood vessels. If there's an interest in inflammatory bowel diseases, then inflamed intestine is often available alongside the tissue that's necessary for diagnosis. So there are good reasons to make better use of these, and I'd say that out of this list, probably the only area which has really made inroads into successful use of tissues is cancer, and it's made the most inroads in terms of also patient education about the value of this tissue. And I would expect that when cancer patients are spoken to, that they're probably more aware of the real value of that tumour tissue to future medical research than, say, someone who has surgery for inflammatory bowel disease. They would probably not be foremost in their thoughts, and obviously they will have 1,000 thoughts in their head at that point in time, but it would probably not be foremost in their thoughts to consider the value of that tissue to future treatments of the disease. So in the next couple of slides, I want to show how this tissue can actually be used and try and demonstrate that I believe it can make a difference. And in very broad terms, there are two things that having a living, functional tissue can do. We can observe the tissue as it responds to a drug, and so basically measure the biological phenomenon, whatever that is, as it happens, and we can then compare the effects of the new drug to an established drug, whether that's a drug with known side effects or what was previously considered the best-in-class drug. So here's a simple example of a test that we do with isolated blood vessels. This is a small blood vessel about the diameter of a human hair. So this is another aspect of what we've tried to do with these sorts of assays, is if there's only a small piece of tissue available, then it's important to miniaturize the test. And so from a blood vessel about 100, 200 micron diameter and maybe 1 to 2 millimeters long, we can get tests of all sorts of conditions. In this example, we have a simple test of blood pressure. So you can see that this is a small blood vessel. This is a cannula perfusing the blood vessel. There is another cannula at the other side which is not quite visible. And you can see a drug has been introduced inside the vessel to cause constriction, and then a second drug to dilate the vessels. So this is an actual measure of blood pressure change in vitro in a human test system. And what I'll show later on is that what happens here is not necessarily what happens in animal blood vessels. A second, maybe more dramatic example is that from heart tissues, there are a whole range of tissue types that can be taken from, in this case, an example of the transplanted tissues that Bob mentioned, such as atrial trabeculae. These are the fine muscle bundles within the atrium, strips of ventricular muscle, or the coronary arteries that you can maybe just make out over the surface of the heart. And I should have maybe put a warning of the gruesome nature of this slide before I put it up, so apologies. But as we're all familiar with human tissue, I thought it would be okay. The second image here shows, again, this miniaturization, trying to remove a tiny strip of muscle, and also you probably can't make it out as it's so small, a very small coronary arteriole, to be able to investigate different functions. And the most amazing thing about this is that the tissues that are obtained often will beat spontaneously. As we heard earlier, transplanted tissues in particular are really handled with a lot of care. They're treated as if they may be used for transplant, and then if it's not suitable or not possible to use them for transplant, they are in prime condition for testing new drugs. So this is one heartbeat from a piece of cardiac muscle, and this characteristic response is essentially the same as it would be in the body. So you can expose the heart tissue to a drug and see whether that speeds up, slows down, increases the force of the contraction. So it's a really relevant endpoint. And what I would also say is that the sorts of measures that you get from here are not that far away from the clinical measures that can be made of ECGs and so on. So just to finish off, this is a graph of a dilatation of a blood vessel. So this is relaxation or dilatation here, so 100% dilatation of a blood vessel with increasing concentration of a drug. This is the normal response that if we took a small biopsy of a blood vessel from anyone in here, we would typically have this response. This is the response that we observed, the upper line here. So this pointer is not working so well. The upper line here is the response we observed in the presence of a test drug, where this same experiment had been conducted in a rat test system in vivo, and there was no effect. So it was later discovered, when they went back and investigated the reasons for this, it was because of a protein only expressed in humans. And this next graph is even more stark demonstration of the differences. This is a comparison of human and canine coronary arteries. The canine test system that's something that's used quite commonly in safety tests of drugs at a late stage. It can prove valuable to prediction of certain markers, but for in this particular instance we have responses. My final point is the three most important things, in my opinion, that can be done is, first of all, provide clear evidence of predictability, and that's incumbent upon all researchers to gather that. And of course the bill was the first attempt to try and address that in a cohesive way. Availability that we're talking about today, there needs to be this bottleneck addressed if ultimately there is going to be increasing emphasis, and there is already increasing emphasis, but even more so a regulatory aspect to all of this testing. So I'd just like to finish off by thanking everyone for their attention and look forward to questions later.